Okay, time to pick back up with enzymes. So what is an enzyme, you might ask? Well, the easiest answer is that an enzyme is a protein that is heavily involved in chemical reactions. Every chemical reaction in the human body, in really any biological system, whether it's a single cell or an extremely complex multicellular organism, every chemical reaction is coupled to an enzyme. An enzyme is so useful in chemical reactions because it is a catalyst, right? What does a catalyst do? Well, it speeds reaction rates without being consumed or altered. And we use the word enzyme to refer to a biological catalyst. A catalyst can really be almost anything. Lithium can be a catalyst. So that is an inorganic molecule. So we sometimes refer to biological molecules that serve a purpose with a special name. In this case, the biological catalyst is an enzyme. So how does an enzyme go about increasing reaction rates? Well, enzymes decrease activation energy needed, and that speeds up reaction rates. Whoa, I didn't explain activation energy. What am I doing? Okay, so activation energy is the concept that every single chemical reaction there is requires energy to start. And that may vary in the amount of energy depending how volatile the reactants are or how stable the reactants are. So a reaction that involves some very volatile components might not require very much energy to activate the reaction, but it still requires energy. You have to take a lighter to a mixture of hydrogen and oxygen in order to start the reaction. So you have to contribute thermal energy to that sucker to start it, even though that's an explosive reaction. So very volatile. And at the same time, if you're going to build a chemical, if you're going to synthesize a protein or a steroid from something simpler, you have to use energy to put the components together. So the activation energy for synthesis reactions generally a bit higher. So an enzyme decreases activation energy of a reaction. So we'll get sort of abstract here. Say you have the reaction, you're building, a plant is building starch to store excess glucose that it has on hand, right? Well, say that without an enzyme, the chemical reaction that builds glucose into starch requires 10 energy. And the plant cell, every second has 20 energy to spend. So how many times can it build starch? How many times can it bind glucose together in a second? Twice. 20 energy costs 10 to put glucose together towards making starch. So your reaction rate is 2 bonds per second. That's kind of crappy. You add an enzyme and it decreases the activation energy required for this. And suddenly the energy cost drops to 2. Your activation energy is now 2. So the cell, with its 20 energy per second available, can then do that reaction 10 times. So your reaction rate goes from twice per second to 10 times per second. So an enzyme doesn't make a reaction occur faster. The reaction that produces the bond still takes the same amount of time to go off. But it allows the reaction to occur more often in a given time. And so that is the reaction rate, how often the reaction occurs. Now, all enzymes are specific. What does specificity mean? What does it mean to be specific? Well, enzymes can only bind to specific reactants. An enzyme has a binding site on it called the active site. Now, you remember how we talked about proteins having to coil and bend and fold into a specific shape in order to function as a protein. Well, part of the enzymes bending and coiling into a specific shape involves the active site being bent and coiled into a very specific shape. And that shape binds to the reactants in the chemical reaction it catalyzes. What that means is essentially you have one enzyme per each type of chemical reaction. Each type of chemical reaction is catalyzed by its very own enzyme. And so enzymes are specific to reactants. You can think of it as the enzyme as the lock and the substrate as the key. The substrate is our fancified science term for the reactants. So the reactants in the chemical reaction are the substrate. So each lock only has a specific key that fits it. So each lock is for a specific key. The enzyme is the lock and the substrate is the key. So enzymes are thought of as a lock and key system. So again, one type of enzyme per type of reaction. Only the right key can fit the right lock. Okay, and just to review, the substrate is the reactants, and the active site of the enzyme is where the substrate binds the enzyme. So how do you go about catalyzing a reaction? Well, the very first step is that the substrate binds at the active site. And then the enzyme catalyzes the reaction, and then the substrate is converted into the products, and the products are not the same shape as the reactants, so they're released. This leaves the enzyme with an open active site and ready to start all over again. And so it's a cyclical process. The enzyme binds to the substrate. Substrate undergoes the chemical reaction. The products are released, allowing the active site to be available so that a substrate binds to the active site. And then the chemical reaction occurs. And then the products are released, opening up the active site to bind to a substrate, which binds to the active site. And then the chemical reaction occurs. And then the products are released, opening up the active site. Okay, I stopped. But you can see it's an endless cycle. And the reason is that an enzyme is not consumed during the chemical reaction it catalyzed, and it's not altered during the chemical reaction it catalyzes. So each time it finishes with a reaction, it's ready for the very next one. So after the products are released, the active site is empty and the enzyme is ready to go again. And a single enzyme can work on 1,000 or more substrate molecules per second. So enzymes are very, very important. Here's an example of an enzyme called sucrase. You'll see that this enzyme ends in ASE. So the letters ASE like that are a dead giveaway because every single enzyme that I am aware of ends in ASE. Just period. It ends in ASE. So if you see a word that ends in ASE, even if you don't know what that enzyme does, you know that it is an enzyme. Furthermore, you might actually know what that enzyme does because they are named most often by either what they bind to or what they do. So in this case, sucrase has an active site that can bind to sucrose, table sugar, made of a glucose and a fructose bonded together. That's a sucrose. So there's your basic rule for sugars. If the word ends in O-S-E, that is a sugar. So sucrase acts on sucrose. Lactase acts on lactose. DNA helicase is an enzyme that unwinds the helix of DNA. So that's named after what it does and not necessarily what it acts on, although kind of both. It unwinds the helix, hence the helicase part, and it does that in DNA, hence the DNA part. So DNA helicase. Sort of a different kind of naming than your nice free association sucrase, sucrose. So here's step one. The enzyme and the substrate are available for the reaction to occur. So the substrate binds to the enzyme at the active site, forming what we call the the enzyme substrate complex. Enzyme substrate complex, just fancified science terms for the reactants are bonded to the enzyme. So at the active site, the reaction occurs. In this case, it is a hydrolysis reaction of these chemical bonds between the glucose and the ribose. Hydrolysis reactions require water. So that's why we have water entering the system here. Once you break that bond, this is no longer sucrose. This enzyme binds to sucrose, so it releases glucose and fructose, and we get right back to step one, where my active site is available, and there's some sucrase around, or sucrose. So, bam, that easy. All right, sometimes you don't always want your enzymes constantly on. Sometimes you want to turn off an enzyme. For instance, if the reactants in the chemical reaction that the enzyme catalyzes are not available to the cell at a certain time, then you might want to turn that enzyme off. And thus, you're not worrying about the energy it costs to catalyze the reaction. You're not spending energy making new enzymes. The enzyme is turned off, so it's not just taking up space and cluttering up the cell. So, enzyme inhibition is slowing down or stopping an enzyme from its function. So it slows or stops enzyme catalysis. There are two kinds of inhibition. The first is competitive inhibition, and the second is the creatively named non-competitive inhibition. Pretty crazy. So what is competitive inhibition? Well, in competitive inhibition, the inhibitor structure or molecule will fit into the active site of the enzyme. It has the same molecular shape as the substrate. But when your competitive inhibitor binds to the active site of the enzyme, a chemical reaction doesn't occur. The enzyme is taken up. So, say I have my enzyme here and it fits a particular substrate right here, and then the chemical reaction occurs and I release my products. Well, say I have a competitive inhibitor, right? And that competitive inhibitor fits to the active site as well. So here we go, and it goes, and it fits, but when it bonds, nothing happens. And now, if the substrate comes back to this enzyme, it can no longer fit. That active site is taken up, the substrate can't bind, no reaction occurs. So, that's competitive inhibition. Another molecule can fit in the active site, and when it binds at that active site, it does not produce a reaction, and thus the inhibitor that can fit in that active site competes with the substrate or an active site. So if you have a solution that has 50% substrate and 50% inhibitor, depending on the physics of the situation, any enzyme in there would have a 50-50 chance of binding to the substrate or the inhibitor. And they won't bind to all the inhibitors at once. So what you'll be doing is slowing down catalysis of those reactions, because you'll be taking enzymes out of commission over time. So that's competitive inhibition. Now let's talk about non-competitive inhibitors. So in this case, a molecule binds, an inhibitor binds to a site on the enzyme that is not the active site. When it does that, it alters the shape of the active site so that the substrate can no longer bind. So altering the active site's shape prevents the substrate from being able to bind to the enzyme, which prevents the chemical reaction from being catalyzed. So there we go. It prevents enzyme activity. So here is a nice little picture of an enzyme here. This is my substrate, so that makes this my active site. And when my substrate binds, I form the enzyme substrate complex and catalyze the reaction. Well, here's my non-competitive inhibitor over here. And here's my site that's other than the active site. We call this an allosteric site. When the inhibitor binds to the allosteric site, A-L-L-O-S-T-E-R-I-C, allosteric. When the inhibitor binds to the allosteric site, my active site is altered. You can see it right there. The substrate will no longer fit. Bam. So that's it for enzymes at the moment. I'll give you your brief single slide anticipatory whetting of the appetite for membrane proteins. These are proteins that determine the overall function of the membrane of a cell. So we're going to learn all about how they do that in video number 3.